Hi everyone. In this section, we will talk about mutation and breeding. After watching this video, you should be able to tell, firstly, the characteristics of mutation, and secondly, summarize the experiments that can prove mutation is spontaneous. A mutation is the permanent alteration of the nucleotide sequence of the genome of an organism, virus, or extra chromosomal DNA or other genetic elements. The gene mutation and chromosome aberration are two important types of mutation. Please look at the picture on the screen. The gene mutation usually occurs during DNA replication, as this picture shows. The next picture shows chromosome aberration. It refers to large fragment change in chromosome. Mutation could lead to the change of codon and further the function of encoded protein. Herein, I would like to point out two important definitions. One is the viral type strain and another is mutant. The viral type strain refers to the phenotype of the typical form of a strain as it occurs in nature. A mutant is an organism or new genetic character arising or resulting from an instance of mutation. As we know the nature of mutation, we could deduce that, to some extent, the function of mutant would be different from that of the viral type strain. On the next, let's take a look at the characteristics of mutation. Firstly, spontaneous. This means the genetic trait could mutate spontaneously. Secondly, irrelevance. It means there is no relevance between the mutant character and the cause of the mutations. Thirdly, rare. Usually, the probability of spontaneous mutation is about 1 in a million to 1 in a billion. Fourthly, independent. It means the mutation rate for one gene is independent of the mutation rate for another gene. Fifthly, inducible. It means the mutation could be induced by mutagen. Sixthly, stable. It means the new genetic traits are stable after gene mutation. Seventhly, reversible. It means certain trait of viral type strain could occur forward mutation or reverse mutation. The mutation can occur spontaneously. Fluctuation tests demonstrate that in bacteria, the genetic mutation arises in the absence of selection rather than being a response to a selection. Please look at this picture. The experiment involved two groups. One is shown at the left side, while another is at the right. For left side group, the bacteria were divided into many test tubers and transferred to the plate after incubation for 24 to 36 hours. For the right side, the bacteria was directly transferred to the plate with division. If the mutations are induced by the media, roughly the same number of mutants is expected to appear on each plate. In this experiment, highly variable numbers of mutants was observed for the plates on the left side. By contrast, roughly the same number of mutants was observed on the right side. So, this experiment suggested that the mutation was spontaneous. Generally, there are two types of breeding. One is breeding by spontaneous mutation, which is time and labor consuming, while another breeding by induced mutation is more efficient. The induced mutation breeding is the process of exposing strains to chemicals or radiation in order to generate mutants with desirable traits. One important step for induced mutation is selection of mutagen. Bruce Amis developed the Amis test, which is a cheap and convenient assay for mutagens. The Amis test uses the bacteria Salmonella typhimurium to test for mutagens. If the mutagen is able to transform his strains into his positive strain, a higher number of revertants would be observed in the plate, adding mutagens relative to the control plate. Finally, let's take a look at the keywords in this section. Okay, that's all for this section.